So at the FENS 2020 meeting this year, I was lucky enough to be asked to give a special lecture for the host society, the British Neuroscience Association. And my talk was about glia-neuron interactions in synapse degeneration and Alzheimer's disease, which is a pretty exciting topic at the minute for me. So we know that Alzheimer's disease, the closest correlate of cognitive decline in terms of brain changes is the loss of synapses. And I've been studying why that happens and how that happens for many years now. And I'd always before been focused really on the neurons themselves and the synapses and the damage to the synapses from the pathological proteins. But over the past few years, a lot more data has been coming out that many of the risk genes that increase your risk for Alzheimer's disease are expressed in glia. And we have found that one of those, ApoE4, and actually two of them, another one called clusterin, that are heavily expressed in glia, also accumulate in synapses in Alzheimer's disease and correlate with accumulation of toxic oligomeric A beta and synapse loss. So in this talk, what I was doing is showing that not only do glial expressed proteins end up in synapses, but the glia themselves seem to play a role in clearing synapses in the brain. So we observed both microglia and astrocytes contain synaptic proteins in human postmortem brain. And in the microglia, we've gone a little bit further and we've cultured microglia either from adult mice or from human resected brain tissue, which was a really rare opportunity and something new for us. Patients undergoing surgery for epilepsy or brain tumors had bits of their brain removed for medical reasons, and the part that wasn't the tumor, for example, would have been thrown away but was given to us. So we were able to isolate living microglia from an adult human and then feed those living microglia synapses from Alzheimer's patient brain or control brain. And the microglia eat the synapses that we tagged with a Frodo so that they glow when they get internalized and phagocytosed. And they ate the synapses from AD patient brain a little bit more and a little bit faster than those from control brain. So that's some direct evidence that the cells themselves can phagocytose synapses. So that was sort of the crux of it, is we're trying to bridge the knowledge gap between these risk genes that we know are expressed in glia and the brain changes that we know correlate with dementia. And so far we have little hints of evidence that one of the things these risk genes might be doing is influencing the phagocytosis or the eating of synapses by glia.